Good day, class, and welcome to our lecture, chapter 8.2. This is going to be cellular respiration, like you've never learned cellular respiration before. I'm going to go over this in a PowerPoint lecture. I will also go over this in a video I'll post for you guys later this week, where I'll draw it out. I think it really helps to write this out as we go through all the steps of cellular respiration. I think it helps to draw them out. So next week I will post this, which we're going to do glycolysis today. Next, we will do the link reaction and the Krebs cycle, followed by the electron transport chain. First part, this is actually from chapter 3, that you should be able to state that in cellular respiration, glucose in the cytoplasm is broken down by glycolysis into pyruvate, and you get a small yield of ATP. You yield 2 ATP. As we move into chapter 8, I want you to be able to outline the process of glycolysis. So this is going to include phosphorylation, lysis, oxidation, and the formation of ATP. There's a lot of vocabulary coming up, so be ready to pause, be ready to write it down. Okay, just a little overview of the first step of cellular respiration is glycolysis. Lysis means to split. Glyco is sugar, so it technically just means to split sugar. And so if you remember right, in photosynthesis, which we'll do next, carbon dioxides are fused together, basically using the energy from the sun in something called photophosphorylation. We make glucose, and then there's a lot of energy stored in that glucose from the sun, and by breaking that glucose apart, we can harvest that energy back out to be used in our cells. It occurs in the cytoplasm, so sometimes you'll call this the TCA cycle. There's a couple different ones because this occurs in almost all living organisms. Bacteria do glycolysis. We do glycolysis. There's no need for a mitochondria. It's just the first step. Your cells are normally doing glycolysis and only really kick in the mitochondria when they're doing something extra work. Does not require oxygen, so if there's no oxygen in the air, you still do glycolysis. But if there is not oxygen, you end up with a final product of lactic acid in humans, as well as some bacteria. And then if you're a yeast, you actually end up with alcohol. So this is how we make booze and hand sanitizer. What happens is one glucose becomes 2 pyruvate. Glucose is a 6-carbon molecule. Pyruvate is a 3-carbon molecule. So you split that in half, you get 2 pyruvates. And it's 10 steps, each mediated by its own enzyme. And so this is a metabolic process. Remember, each step is mediated by an enzyme. And very true in this one, pyruvate can actually act as an end product inhibitor of the first enzyme in this step. We'll go through what that is. You do not have to know any of the enzymes. Just know that it's mediated by enzymes. Overall, you produce 4 ATP, but we have to put 2 ATP in. So you only get a net gain of 2 ATP, 2 NADHs. You only have to know that as NADH. It's actually nicotinamide. It's something, I can never remember it. But you get 2 ATP, 2 NADHs, and 2 pyruvate. This is a must know for all of you. The next part's the higher level. So if you want to score really well on your test, especially in this class, you're going to have to learn this stuff inside and out. Before we get into how glycolysis works, we got to do some vocab words. If you guys have had chemistry, this becomes much easier. If you haven't, it's much harder. So we have 2 words that we're going to use a lot as we describe things, and it's going to be oxidation and reduction. So oxidation, think of iron. When iron rusts, it's oxidized. It's a redox reaction. So these are redox reactions, reduction, oxidation. So what happens there is iron gains in oxygen, so we call it oxidation. And it's going to be actually what happens here. So what is oxidation? It's the partial or complete loss of electrons. So when electrons are lost from a molecule, we call that oxidation. When that happens, the molecule actually gains an oxygen as well. So it's the gain of an oxygen, loss of electron. So here just goes through the major things that are in an element, and during oxidation, electrons are lost, hydrogens are lost, they normally go together, oxygen is gained, and overall, we would say the molecule has lost energy. So that energy has gone into the reduced molecule, which is our next stop. So reduction is the gain of electrons, often in the form of hydrogen, also probably means the loss of oxygen. So electrons are gained, hydrogens are gained, we gain more energy, and there is a loss of oxygen. This sounds like a lot now, but we'll go over it again when we get through how glycolysis occurs, and hopefully that'll help you understand it more. But I would definitely write down this chart. I did not put it in this PowerPoint, but a good saying that helps you remember this is oil rig. So oil is oxygen is lost, okay? Rig is reduction, energy gained, or is gained. So oil, oxygen is lost, rig, reduction is gained. So, and we're talking about the electrons though, there. So if you remember oil rig, rig has a G, it means it gains an electron. Oil has an L, it loses an electron. We'll go over that again, but it's pretty helpful. Next big vocab word that we're going to use a lot is something called substrate phosphorylation. And this is the process of adding a phosphate group to something. This is one of the first steps actually of glycolysis shown here. And we're going to take a glucose and using a kinase. So kinases are going to be enzymes that typically transfer a phosphate group. So in this case, two names this is given. I always call it a hexokinase because it's a 6-carbon sugar, but you could call it a glucokinase because it's working on glucose. And what happens to this is an ATP comes in, phosphate is popped off the ATP to make ADP, and then that enzyme attaches the phosphate here. So this would be a type of substrate phosphorylation. We'll look at another type where you can actually add a phosphate to an ADP, and this kind of goes backwards. Still would be substrate phosphorylation. So you're using a kinase enzyme to add a phosphate to something. For your purposes, we'll just keep it as like adding it to a molecule using an enzyme of sorts, normally from the power of ATP, but sometimes you actually make ATP. Okay. This next one, you don't have to write down anything on this right now, but this is the beginnings of glycolysis. This is what we're going to learn. And so here is glucose and that hexokinase, and you're getting that ATP is coming in, it's being used, and we're phosphorylating glucose. So now we have this glucose with a phosphate group, and this actually happens twice. So we end up adding another phosphate, and you get two of them. And then this guy switches, and you can see all these little enzymes are mediating the whole process. Once again, we're going to get something called NADH made here. This is what we would call a reduction of NADH, okay? Because it's adding an electron, adding energy. We're oxidizing this molecule that was right here. And then we form these two molecules. At this point, there's two, because remember, we've broken the carbon up. I'll go through this much more in depth. But then we get 2 ATP out of this one, and we get 2 ATP out of the other one as well. So you get 4 total ATP and 2 pyruvate. You don't have to know it this complicated, but I kind of just wanted you guys to see that it is a very complicated process, and we're just going to make it much simpler to describe today. Okay, and it was also my examples of substrate phosphorylation. So this is substrate phosphorylation. We're taking this phosphate and we're adding it to this ATP using a kinase, okay? So that is substrate phosphorylation. We're going to also talk about oxidative phosphorylation here in a minute. Okay, so here is our oxidative phosphorylation. We're not going to get to this till the end of cellular respiration, but it's the production of ATP using energy derived from the electron transport chain. So instead of using a kinase now, we're going to actually use the energy of a proton gradient to turn an enzyme called ATP synthase, and this is a very special type of phosphorylation called oxidative phosphorylation. We will learn a third phosphorylation, which is photophosphorylation. photo meaning because the energy to do this ATP synthase is going to come from the sun, and it actually uses an electron transport chain too. But in oxidative phosphorylation, the energy to make the protons is from the oxidation of NADH and something called FADH. So that's the energy comes from oxidation, where here the energy is going to come from this kinase and from the phosphates that are here. And technically the energy came from ATP in this step. Okay. Just to give you an idea, 90% of our ATP is made during oxidative phosphorylation or the electron transport chain. But we're going to start to try to use this term oxidative phosphorylation instead of electron transport chain a lot. This is also known as, coming up, chemiosmosis. Oh, 3 different things mean the same exact thing. Pretty crazy. Okay, we talked about these NADHs that I've talked about and NADs. They're called electron carriers. There's actually an FAD that we'll talk about too. You do not have to know that it's nicotinamide adenine dinucleotide. You don't need to know that, but basically it's like a special form. Here, I have a picture of it. It's like a very similar to a nucleotide, but they're very special nucleotides that have that carry electrons. Very cool. Actually, ATP is a nucleotide as well. If you haven't figured that out yet, we will as we get into how DNA is made. So NAD and NADH are basically the same things. This is the oxidized form, and this is the reduced form. So NAD is oxidized form, NADH is the reduced form. And why they're important, they easily accept electrons and a hydrogen to go with it. That's why we add the H here, is because there's a hydrogen. They're just really good at picking up hydrogens and carrying them, and then they can release those hydrogens and electrons really well, and they carry a ton of energy. I mean, they are like right up there with ATP and energy. They're pretty amazing. Actually, 1 NAD can produce 3 ATP. So there is a lot of energy here, but it's not as easily used by the cell as something like ATP is. So ATP is easier to use. This is like carrying a nuclear bomb. Okay, big key things here. NAD is reduced to NADH. We're going to get used to saying that. So NAD is reduced to NADH. So this is our reduced form. And then vice versa. NADH is oxidized to NAD. Okay, so oxidized, meaning it's going to release its electron, release its hydrogen in the form of NAD is what we get after that. Okay, so real quick, let's go through glycolysis. Like I said, I'm going to do this again, but what glycolysis really is, it's the oxidation of glucose to 2-pyruvate, which is a 3-carbon form, and it's coupled with the reduction of ADP to ATP. Wow, we just used those words, oxidation and reduction. So when I oxidize glucose, I'm taking electrons away from it, right? And we end up with 2-pyruvates that have lost some of the energy because they've gone to electrons, electron carriers, or ATP. And then we also have this reduction, so we've added energy to this ATP. This would be substrate phosphorylation as well. Here's a very simple way of writing it, glucose to pyruvate, but you guys just saw the whole steps that actually go through this. It's much more complicated, but really glucose is oxidized to give us 2-pyruvate, and from that oxidation we get a reduction of ADP into ATP. Let's break glycolysis into 3 phases. We're going to have an energy investment phase, so we're going to put energy into it in the form of ATP. We're going to have the lysis phase, where we're going to split glucose. And then we're going to have the energy harvesting phase. So we're going to take those phosphates that we've added to glucose, and we're actually going to produce now 4 ATP for a net gain of 2. And that is how we do it. So here is what happens in it. So we have our 6-carbon glucose. This is what I'll draw in a lecture later. We have a 6-carbon glucose, and we're going to invest 2 ATP. Remember, a little kinase is going to come in, and this is about 4 steps, but we're going to simplify it to 1. So we go from a 6-carbon glucose to adding 2 phosphate groups onto this glucose, and this has now become fructose biphosphate, or you could just call this a phosphorylated 6-carbon sugar. I would be fine with that. And then that is quickly split. It actually becomes very unstable, and it is split into 2 molecules we're going to call G3P. So this is a triose phosphate. So there's triose for 3 carbons, and on the 3rd carbon we have a phosphate. So we have 2 triose phosphates now, and then from that step, they're going to be harvested for their phosphate group right here, and we're going to net 4 ATP out of them. So each G3P gives us 2 ATP. Since there's 2 G3Ps, we get 4 ATPs. At the same time, we also have the reduction of NAD. So NAD is reduced, this G3P is oxidized, and we get NADH. So NADH now has taken the electrons and energy from this G3P. ATP has taken some of the energy from the G3P, and we now have 4 ATP and 2 NADHs. We're just going to kind of put these NADHs in our back pocket because we're going to use them in the electron transport chain. The ATP now, we can just go use for whatever we want to use ATP, like a pump in your muscles, sodium, potassium. There are just all kinds of places to use that ATP. So we're going to break it down, do this so many times because I know it is very complicated. So energy investment phase, glucose phosphorylated twice, okay? And we get this 6-carbon phosphorylated 6-carbon molecule right here, fructose biphosphate, if you want to know what it really is. I don't care if you call it that. Okay, this is these 3 sides. This is what you really want to write down, okay? Energy investment. Glucose is phosphorylated twice to form a 6-carbon phosphorylated molecule, okay? Sugar. Lysis, that means we're going to split it. So that molecule is now split into 2 G3Ps, triose phosphates, 2 G3Ps. You're going to get used to these. They come into play again in photosynthesis. So we got these 2 G3Ps, and then those G3Ps are going to be harvested for their energy. So at this point, another inorganic phosphate comes in. So we actually end up with this 2-phosphorylated 3-carbon molecule. And at that point, we're going to oxidize NAD to NADH, okay? Or no, I'm sorry, we're going to oxidize G3P and reduce NADPH. You can just say reduce NADH. So that's going to be some energy we use later. And then the next phase, we actually generate ourselves 2 ATP. So we knock off each of those phosphates, add them to an ADP, and we got 4 ATP, 1, 2, 3, 4, and we are left with 2 pyruvates. That is glycolysis. If oxygen is present, this pyruvate is going to enter into something called the Leak reaction, which we will do next. If no oxygen is present, you guys talked about this before, this actually changes into lactic acid, which is a 3-carbon molecule as well. No CO2 is lost. It's pretty nasty stuff. In yeast, we actually chop off one of these carbons and it becomes CO2. Ooh, we get bubbles in beer, and we get ethanol, which is a 2-carbon molecule. Okay, so here it is. Just again, that's that second part, different diagram. We got the 6-carbon that's got 2 phosphates on it. It is split, lysis split into 2 G3Ps. Then that is harvested where we get 4 ATPs, 2 NADHs, and we end up with pyruvate. Okay, in all, this was all done through substrate level phosphorylation. And basically, it was created by that ADP bringing in a new one to form ATP. That is our energy molecule. To sum it all up, each molecule of glucose is broken down to 2 pyruvates. We net 2 ATP. We also net 2 NADHs, which remember are reduced energy. They're going to carry lots of energy, and we're going to use those NADHs to eventually make more ATP and the electron transport chain. That was a lot. I would try to go back and outline the process. Like I said, I'm going to do this also handwritten on the board in a videotape for you that I will post probably by Friday. Then we'll jump into the other two sections as well. You guys have a good day. Don't forget to answer your questions.